Hello guys, I hope you guys are all doing great. I'm doing actually another video um, right now about the book that I bought on aging. I think it's a very nice book, so I wanted to talk a bit more about it. I talked briefly about it in the last video, just as an introduction. But I want to start by talking a bit about cancer. And so in the 1960s, right, it was simply the way it is. People would die of cancer, and it was actually a very tough battle for many patients. Well, in the 1970s, right, there were some genes that were discovered, like BRAF2, HER2, and BCR-ABL, as well as immune therapies, which will literally bind to cancer cells, making them visible to the immune system, specifically to the T cells of your body. And so aging today is similar to the research we had on cancer in the 1960s, whereby we were like accepting it kind of the way it is. But yeah, and actually until the second half of the 20th century, that's how it was. It was the natural way. That's how it is. So, so there were some theories about why we age, and Haldane and Medawar and Williams, so the trio, right? So these three people actually said it's due to something called a selfish gene being passed on. And in the 1970s, that was kind of restated by Kirkwood, which talked about the disposable soma hypothesis, where those who breed fast die young, and those who breed slow maintain their soma and live longer. Other than these evolutionary theories, others include DNA damage. You probably heard of this. Simply, aging is due to damage to your DNA, and that was proposed by Medaware and slizard in the 1950s and 60s. And then also error catastrophic theory in 1963, which says it's based on mutations to our cells when the DNA replicates, right? So error catastrophic theory, so cells accumulate errors due to cell division, due to DNA replication. And then Denham Harmon stated it's actually due to free radicals, which activates our natural defenses. It's actually a $3 billion industry right now. Drink this, drink that. An interesting thing that came out in the 1960s, slizard actually initiated cloning that showed that old clones can actually live normal lives after nuclear transfer, which disproves DNA damage as the main reason, because even with the old DNA, you can still get a completely new healthy organism. And so therefore, there's actually 7 reasons of why we age. I spoke about them in the previous video, so I won't do it. But it's not just DNA damage. It's, of course, more comprehensive. There's definitely much more to it, right? Many reasons, like issues with the mitochondria, senescent or zombie cells, etc. So the human genome was actually, there was a human genome project in 2003, which was mapped. And it shows how little we know, because essentially dark matter is 69%. And even of everything that we decoded, we still don't know 10% of it, of all of the coding genes. So 69% is just non-coding junk. And so that just shows. And then there was this guy called Mortimer who did a yeast research and caused it to be blown. Many people then experimented with the yeast. And David Sinclair actually moved to Vienna to meet Guarent, which was the top scientist in yeast research at Harvard University in Boston, Massachusetts, where he studied Werner syndrome, which is present in individuals with premature aging. And that actually later led to him discovering the SGS1 gene in yeast, of which mutations would cause essentially the nucleolus to explode and prevent the ability for the yeast to mate. So that cause sterility. The yeast will lose fertility. Essentially, the way it works is broken DNA causes genome instability, which then distracts some genes called sirtuins, specifically sirtuin 2, which was first discovered in yeast, which causes the change in the epigenome, causing the cells to actually lose identity and become sterile, while trying to fix the damage at the same time. So you actually get these ERCs, or extra ribosomal chromosomes, which then become larger and larger as they multiply, and then they're reinsert into genome, excised. They're like retrotransposons, and essentially you get these big circles in the genome. Oh, that's not good. And so the sirtuins try to go and repair them, but as you do that, you get these epigenomic changes where the cells literally lose their identity and don't act like themselves anymore, and that leads to aging, disease, and death. And this is similar to a piano key in a chord missing keys or playing extra keys, which just doesn't sound as good, to the point where you want to cover your ears because it's the symphony is just horrible. So this loss of identity is caused by epigenetic changes, and this actually leads to cells losing identity and then becoming senescent. So they essentially stop dividing. And this senescence is what leads to aging, right? Cells don't replicate as much, you get organ failure, etc. So these ERCs, these genomic instabilities, cause sirtuins to move to the nucleolus, actually. And so that's why you see the nucleolus, and it dies in the microscope. That's how what they notice taking place, and that actually leads to senescence of these yeast. And that was published as the cause of aging in 1997. And it was actually shown that a low sugar diet causes less ERC accumulation, which causes less breaks, which causes less nucleolar export, which leads to the avoidance of sterility, disease, and death. So essentially, the low sugar diet is a good way to go. And as you'll see later, and I already talked about it, fasting in general is a good way to go. Eating less sugar, eating less amino acids or protein. And then just to confirm that, in 2003 and 2006, studies literally showed that the lower you have levels of sirtuin 1 and 6 causes lower lifespan. And in 1999, it was shown adding more copies lengthens the lifespan. So the more sirtuin you have, yes, you're going to go fix the damage, but some of it is going to remain in that site to prevent the epigenetic changes that are taking place, allowing cells to maintain their identity, right? And so that's very, very important. And it was actually later shown in 2017, actually recently, a couple of years ago, in humans, that what sirtuins do is actually they stabilize this ribosomal RNA to prevent senescence, and that's how they extend their lifespan. So the 2003 and 6 studies, I believe, were in 1999 ones, I believe were done in mice, right? What they have published was done in yeast, and then recently was actually shown in human cells. I believe in a petri dish, if I'm not mistaken, but still, nevertheless, human cells. And so these sirtuins, yeah, they leave their sites to address the DNA damage, to fix it, and that's what causes the epigenetic changes and loss of cell identity. So an experiment was then done, actually from slime, and from slime they injected certain plasmids. The plasmids that come from slime, it's like IPPOL9. These plasmids, they're called ICEs, which induce change to the epigenome. And what happens is they add these plasmids, and then they add a drug called tamoxifen. And when these plasmids are present in the presence of a drug, tamoxifen, they cause a cut in the genome. And because tamoxifen has a short life, or a short half-life, so to speak, they can quickly change or reverse the effects or the changes. And so when they cut the genome due to tamoxifen, mice aged faster. Why? Because you're inducing the DNA damage, you're causing genomic instability. And you literally saw all of the changes of aging, like low muscle, bone issues, osteoporosis, poor vision, glaucoma, low mitochondria, so fatigue, they have low energy, dementia, and frailty. So you literally get an aging mice. Now, there's actually just in nature, right? There's many animals that live eternally or for very, very long, like bristlecone pine trees, Hydra vulgaris, which is like a jellyfish that can regenerate its body parts, pretty cool. Greenland sharks, which ages very, very slowly or not at all, and it literally does not does not have sexual maturity by 150. So again, longer to breed, fewer offspring, longer lifespan. As well as the bowhead whale, which is very cool because this whale, again, less predators, takes longer to breeds slow, fewer offspring, less predation, right? And that's good because we actually share a lot of our genes with this bowhead whale, so we can study it. And we actually share almost 13,000 genes, like 12,700 and something. And have these FOXO2 or DAF16, which is identified by Kenyon as a longevity gene found in these whales. And we actually have 4 of them as well: FOXO1, 3, 4, and 6. And in yeast, there's a variation called MSN2, which turns on genes that recycle NAD. So this FOXO3 present in these whales actually is also found in places where people live very long, like in the China Red River Basin. So what happens is this FOXO3 gene is found in these very long living whales that live pretty much almost forever, and they actually have this gene FOXO3, which keeps this NAD level up. We'll talk about NAD later on. But that's kind of what Waddington talked about. So essentially, these epigenetic changes cause a change in the cell identity. And so if you look at differentiation and said that just like marbles, which get jostled from the top of a mountain to valleys, which cause the ball to fit in various grooves due to gravity, assuming various positions, right, because of different grooves and cavities, so does our body due to DNA damage. Sun, X-rays, all that causes a change in our identity, and therefore a loss in the functioning of these cells. Therefore, we need to keep these values strong, or very vertical, very steep, and gravity strong. And so how can we do that so that cells maintain their identity? And we already saw in mice published in 1997 exactly what was happening with this extraribosomal genomic instability. Essentially, you can do it by adding sirtuins. 1999 shown that you add more copies of sirtuins, right? You actually extend lifespan. And in fact, it was confirmed in humans just recently. As well as adding NAD, as well, really helps. As I mentioned, I think in the last lesson, or in the last video, sirtuins need NAD to function. So that makes sense that if you increase NAD levels, and actually we have these FOXO genes which recycle this NAD, which is important in long-lived whales, right? That's another way that we can actually allow that to happen, right? Keeping those valleys vertical and the gravity strong for the cells to maintain their identity, so the cells don't lose their identity, so they keep functioning properly. And so in the next videos, and I already mentioned it briefly in the previous, we'll talk more about the natural ways you can do it, as well as some pills that you can take. But some natural ways, to your surprise, is fasting, as well as high intensity interval training. I think I alluded to that in the last video. And so with that, I will see you guys in the next video, where I will talk about these natural boosters that you guys can actually do right now at your home to actually live longer. And one of them is, you guys not gonna like me for that, but stop eating 3 meals a day, or essentially eating less of a portion size, while ensuring that you don't undergo through malnutrition. So still eating properly, but just a bit less, not overeating. In fact, eating considerably less helps promote lifespan and live longer, as well as intensity workout, vigorous intensity, like going like around 70 to 85% of your maximum heart rate, essentially taking your breath out, as I like to say, going for a heavy jog or something like that, right? Going for a fast run where your heart is beating fast and you're barely able to talk, all right? So we'll talk about these natural ways in the next video. I hope you guys have a great day, and I wish you all the best, especially in this difficult time of the pandemic. I hope you guys are all doing very, very well, all right? Peace. All the best, you guys. See you in the next video.